Good afternoon. I'm Dr. Karen Antman, the Dean of the Boston University School of Medicine, and we would like to welcome you to a terrific conference on neuroscience. The panel will be moderated by Dr. David Greer, who is our chair of neurology at Boston University School of Medicine since 2017. He studies recovery after coma, after cardiac arrest, acute stroke treatment, and stroke prevention. He received a BA in English Literature from Williams College and a master's in English Literature and an MD from University of Florida. He then completed a neurology residency and fellowship in vascular neurology and neurocritical care, both at the Massachusetts General Hospital. He stayed on as faculty until 2010, where he advanced to an associate professor of neurology at Harvard Medical School. He then joined the faculty at Yale as a professor and vice chair of neurology and the director of the stroke program before being recruited to Boston University School of Medicine. He's authored more than 250 peer-reviewed manuscripts, reviews, chapters, guidelines, and books. Dr. Greer?
Great, thank you, Dean Antman. It's a real pleasure to be with you today, and thank you everybody for joining us. It is a really exciting moment for us to be able to share with you what's going on in neurology. I think it's been nothing short of revolutionary what's happened for us in the last few years with great developments in our clinical care, our education, but also the research, which is what we're going to highlight for you today. We have some true centers of excellence in the things that we do in neuroscience research at our department. And we're going to showcase a few of them for you today. I'm sure everybody's already heard about the Chronic Traumatic Encephalopathy Center at BU, but this is really world famous, led by the famous Anne McKee and Bob Stern, but we have other wonderful investigators there that you're going to hear about their work today, including Mike Alasko and Jesse Mez. We also have the Framingham Heart Study, for which the neurology department does the majority of the neuroscience work in that area, including stroke, but also dementia, movement disorders, epilepsy. So really a tremendous 70-year population-based data that's a gold mine for what we do at Boston University. We also have a continuously NIH-funded Alzheimer's Disease Center since 1999. So going on 22 years of continuous funding for the Alzheimer's Disease Center. Huge public health problem, only going to grow as the population grows. And so we really need breakthroughs in that area. And you're going to hear some of the work that we're doing in that area as well. We'll also highlight the Parkinson's Disease and Movement Disorders Center. Really great work being done in deep brain stimulation, which has been revolutionary in the field to improve the lives of people with Parkinson's disease and other tremor and movement related disorders. We have a really outstanding residency training program now. Our residents are making huge contributions in the field. Most of them are going into academics, many of them into practice as well, and many of them taking on scientific careers of their own. We've just applied this year to have an R25 program, which is an NIH-sponsored educational and research program for residents during training to get a leg up on being a physician scientist. So we're very, very excited about our future in neuroscience at BU. I want to mention our gratefulness to the many donors who supported our work over the years and honor their legacy through the research that their funds support, such as the SPVAC Excellence in Neuroscience Awards, which are extremely helpful for our investigators. This was established by Jack Spivak, as well as the FEM and Victor Hochberg Memorial Fund, which was recently established to support neurological research. We really couldn't do much of what we do without that support, and we are so extremely grateful. So enough of my talking. I want to introduce the panelists. Let me tell you the four, and then we'll start with Dr. Alasko. But to just give you the bird's eye view, Mike Alasko is an associate professor of neurology and the lead investigator of the BU CTE Center. He is a PhD in neuropsychologist, probably one of the most prolific scientists I've ever known. that I couldn't believe his CV when I first saw it. I thought this guy is a total star, and then you meet him and he's also one of the nicest people on the planet. So it's really such an honor to work with him. Next is Jesse Mez, who I actually had the pleasure of working with when he was a resident back at Mass General. He's now an associate professor of neurology and the director of the Alzheimer's Disease Center Clinical Core, and really an amazing scientist and contributor to the field. Ludi Shi is one of our newest recruits. She joined us just last year. She's associate professor of neurology, and she's the director of our deep brain stimulation program. So you'll be hearing some of the very exciting work that she's doing, as well as the rest of the group in that realm. And then Lan Zhao will be batting cleanup for us. She's professor of neurology and pathology and a neuromuscular researcher and muscle pathologist. She also has a true basic science lab and translational science, working with mouse models that she's developed herself. She's also been, as I said, our research vice chair, our first ever research vice chair, and has given her heart and soul to that job to really rev up and make our research in the department excellent at every level. So with that, that's the group that we have to look forward to, and I'd like to introduce Dr. Mike Alasko to give our first talk.
Hi, everyone. Thank you, Dave, for the very nice introduction. I am thrilled and excited to be invited to this event and to interact with you all for the very first time. I've been at BU School of Medicine now for 5 or 6 years, and it's just been such an incredibly exciting time for research and discovery. I couldn't imagine being anywhere else, and it really a testament to BU's people and the resources, including Dr. Greer and Dean Antman as well. So my research, focuses on the study and the detection of the long-term effects of repeated hits to the head, including a focus on chronic traumatic encephalopathy. Repetitive head impacts, what I mean by that are the types of hits you see from playing American football or playing other contact sports like heading in soccer, rugby, but also in military combat, also physical violence like intimate partner violence. These types of repetitive head impacts, they've been associated or been identified as a risk factor for chronic traumatic encephalopathy, where I really spend most of my time. So CTE, it's a progressive brain disease. So we all have this protein called tau in our brain. It becomes abnormal in CTE, and it can lead to problems with thinking, memory, mood, and behavior, and eventually really impair one's ability to function normally. It's similar to Alzheimer's disease, but the way that the tau presents and the type of tau it is quite different and distinct. I'm showing you an image here where the brown is tau and it accumulates around blood vessels, something that's very unique to this disease. It's been diagnosed pathologically in various contexts: sport athletes, American football players, ice hockey players, soccer players, rugby players, and in military veterans as well. A key problem or a key knowledge gap in this field is that we don't yet have tools or the ability to accurately detect it or diagnose it during life. Given the millions of people with this history of repeated hits to the head, this represents a really important problem or knowledge gap that we need to address. And I spend a lot of my time using neuroimaging to address this knowledge gap. So we just launched this study, actually just recently, that we're calling FIND CTE. FIND CTE is focusing on tau PET imaging. So PET imaging, it's a brain imaging technique where a radio tracer is injected into the body and it can travel up into the brain. And this tracer can bind to targeted proteins. In this case, we're really interested in tracers that can bind to tau. So can we use a tracer to pick up that tau pathology we see on the brain at death? There's been a lot of exciting developments, including some at our center on this front, but we still have not yet identified the optimal tau tracer that can pick up on the specific CTE pathology. And I'm working with folks at University of California, San Francisco, and really over the next 2 or 5 years, we're really hoping we can find out the optimal tracer to detect this disease. Beyond tau PET imaging, we're also looking at more routine imaging measures, specifically structural magnetic resonance imaging, or MRI. So memory disorders, things like Alzheimer's disease, every evaluation will involve an MRI. We look at the MRI to look for patterns of shrinkage or death to the tissue. Different patterns can provide clues onto what might be going on, which is incredibly important for us to be able to treat and diagnose the disease. We have some really exciting results where we're seeing on routine MRI patterns that map on to exactly what we see at death when we look at the brain. Specifically what Dr. McKee and her team finds when they look grossly at the brain, we see very similar patterns, and this is very promising research in our ability to detect it. And in fact, our colleagues from radiology at Boston Medical Center, Drs. Goldstein and Anderson, were just awarded a Massachusetts Life Science grant to get us a brand new MRI scanner, which is pictured there in the upper left. This is really state of the art. And we're really going to use this machine as much as we can to unravel what the MRI profiles look like in CTE. In addition, we're also using magnetic resonance imaging to study the white matter. So we all have these long fiber paths in our brain, and they seem to be really susceptible or prone to injury from head trauma. And in fact, we're seeing that years after playing American football, this white matter injury continues to persist and progress. And I'm using some novel software where we can actually pick up the white matter injury, detect it, and also quantify it. And I'm just showing you here some of our recent data where we see actually a direct association between the more years of playing football, the more white matter injury we see on MRI. All of my research is really done in the auspices of the Alzheimer's Disease Center, as Dr. Greer mentioned. Our center is one of about 30 in the country funded by the NIH. Not only does it help us facilitate our own research, but really the goal of this is to provide a resource to the local and really the national and even international scientific community. We follow about 400 plus participants. They undergo annual, very comprehensive, detailed assessments. And actually one of the focuses we've been looking at is, can we detect these different neurological diseases like Alzheimer's disease, like CTE, using in the blood? So instead of these costly, more invasive imaging, can we just get a blood draw and detect these proteins? And can that be a much more feasible and scalable way to assess these diseases? What's really exciting that we have going on, and this is led by, again, Dr. McKee, with me and Dr. Mez also having leadership roles here, is this new Brain Bank Collaborative Initiative. So this is a gigantic effort that's being funded by the NIH, that's being led by the BU School of Medicine. Basically, and we're also collaborating with Mount Sinai as well. And what this is doing is harmonizing 6 different brain banks at BU, as well as brain banks at Mount Sinai. And we're going to harmonize them in order to better understand the long-term effects of TBI, traumatic brain injury, or repetitive head attacks on the brain. What are the risk factors? What are the clinical symptoms? And how can we diagnose CTE in life? This study will address those questions. I just want to conclude by really kind of driving home the point that CTE is a major public health concern. Millions of people play contact sports every year, serve in the military. There's millions of people who are frustratingly having symptoms, and we need to be able to detect and diagnose the CTE in life. Once we can do that, we can then start to open the doors and start to conduct research where the ultimate goal of being able to treat and prevent this really progressive brain disease. If you have other questions, I would love to interact with you more, get advice, and also work together to figure out how we can address this really timely and major public health concern. Thank you so much.
Wonderful talk, Dr. Orlowsko. Thank you so much. We're going to get to the Q&A section at the end. Feel free to put your questions in the Q&A box whenever you'd like, and I will distribute them at the end of all the speakers doing their turn. Our next talk is from Dr. Jessie Mez, who's again Associate Professor of Neurology and the Director of the Alzheimer's Disease Center Clinical Core. Take it away, Jessie.
All right, so can everyone see my screen? So thank you so much, Dr. Antman and Dr. Greer and Dr. Olasko, wonderful talk. And I'm so happy to be here to be able to share some of the research that I'm participating in with all of you. So I, like Dr. Olasko, work closely on chronic traumatic encephalopathy as well as on Alzheimer's disease. And Dr. Olasko and I work very carefully on both of these topics, and I'm going to touch a bit more on both of them. So you can see here that I do have a lot of titles, and I just mentioned that briefly because these are all different resources: the Alzheimer's Disease Research Center, our Chronic Traumatic Encephalopathy Center, the Framingham Heart Study, the Alzheimer's Disease Genetic Consortium. These are all resources that are really unique to Boston University and that I leverage as a way to facilitate different types of research. Each of these different resources offers a unique opportunity, a unique resource to ask a different research question. So a little bit more about chronic traumatic encephalopathy. It's a degenerative brain disease. Like you heard from Dr. Olasko, it's found in athletes, military veterans, and others with a history of repetitive head impacts. For the physicians in the audience, when I was in medical school, we learned about this in our second year pathology class. It was called dementia pugilistica. It was something that you learned about, but whether or not you truly ever would see it in life wasn't clear. And that was before CTE started to be described in football players. Back when it was initially described, it was largely a disease seen in boxers. This has changed in the last 10 years. Our brain bank, which you heard a bit about, is the largest source of brain donors with chronic traumatic encephalopathy in the world. To date, we've now had nearly 1,000 donors, all who have a history of exposure to repetitive head impacts from contact sports, military service, or from physical violence. In 2017, we presented these really striking numbers from the brain bank: 110 out of 111 former NFL players and 48 out of 53 former college football players. And you can see some of their pictures on this slide, and they ask that their family members ask that we show their faces, that it helps raise awareness, and they want the public to know how devastating the disease this was. So my work, like Dr. Olasko's, also focuses on diagnosing CTE in life. So like all neurodegenerative diseases, we can only definitively diagnose CTE after death. For Alzheimer's disease, we're quite good at this point at diagnosing it in life. But with CTE, it's very much still limited to death. And so in order to treat CTE, we need to be able to diagnose it in life. And again, the doctors in the room, on the panel, listening in, know that the most important part of seeing a patient is their history. And especially in neurology and neurodegenerative disease, getting a careful history is crucial to make a diagnosis. And the way that this works in neurodegenerative disease and in neurodegenerative research is that we take a careful history, and we have particular criteria to diagnose the disease in life. And what we are doing as part of our brain bank research is we're going to the loved ones of those who have passed away and donated their brains, and taking the careful history like we would do in the clinic, and then we're comparing that history to the neuropathology. And we do that as part of a clinico-pathological consensus conference. You can see here one of the typical conferences that we have with me and Dr. McKee, Dr. Stern. You can see Dr. Alasko in the background. And we really use the pathology as the gold standard to try to diagnose CTE in life. I'm also really interested in the epidemiology and risk factors for CTE. So what we've learned is that it's less about an individual traumatic brain injury or an individual concussion. It's more about the amount of exposure over time. And we've shown that with each additional year of football play, the risk of CTE increases by about 30%. And that equates to a doubling of risk every 2.5 years. And this is what we're showing on what I'm showing in this figure here. You can see the dark blue are those with CTE, the light blue, those without CTE. Similarly, the dark blue here is those with severe CTE compared to those with mild CTE. In both cases, more play leads to increased risk of disease. We're also very interested in the genetic architecture of CTE. So we can see two individuals who have played almost the same amount of football, one who develops the disease and another who doesn't. And that suggests that there are other risk factors in addition to exposure to contact sports and to repetitive head impacts as a risk, and potentially genetic factors. In order to do these genetic studies, we need to carefully characterize the brains that we're looking at. And what you see all the way to the right is, rather than just looking at those with the disease and those without the disease, we can actually count the number of neurons that have the tau uptake, like you heard Dr. Alasko describe. And we can use this rich outcome as a way to identify genetic associations. We're also really interested in the prevalence of CTE. So I gave you these really striking numbers from the brain bank, but the brain bank isn't really representative of the general population of football players. And to understand how common this disease really is, we need to look to epidemiological cohorts like the Framingham Heart Study. So we are carefully characterizing the amount of contact sport exposure among participants in the Framingham Heart Study. And we're looking at the relationship between dementia, Alzheimer's disease, CTE, and other outcomes in the study. We're also leveraging other epidemiological studies of aging, including the Religious Order Study, the Memory and Aging Project, and the Adult Change in Thought Study in Chicago and in Washington State. And then even when we use those epidemiological studies, we still only see maybe 15 or 20% of the men who have played contact sports or who have experienced repetitive head impacts, and far fewer of the women. So ideally, we would like to look at a population that is large, but also at high risk. And we've recently initiated this new Boston University football study. And right now we're conducting a pilot study. We're reaching out to every former Boston University football player so that they can fill out a questionnaire so that we can get a report of their head injuries and concussions and whether they're having any cognitive, mood, behavior, or motor symptoms. And the hope is that we can leverage this to bring many of these folks in, actually as part of our Alzheimer's Disease Center, to do our annual assessments of cognition, of imaging, of blood and cerebrospinal fluid biomarkers, like you heard from Dr. Alasko, as well as genetic studies. So now I'll switch quickly to Alzheimer's disease. So Alzheimer's disease, we know much more about. It affects more than 5 million Americans. By 2050, this number is projected to rise to nearly 14 million. And this is almost entirely driven by this purple here, those increasing age above 85. And it's the 6th leading cause of death in the United States. And despite more than 100 clinical trials, there is not a therapy that slows or stops disease progression. And the reason why, or one possible reason why these therapies have failed is because we've considered the disease as a single entity rather than perhaps multiple entities. There's not a single type of cancer. Even within cancer, there are multiple subtypes. So perhaps by looking at risk factors and by considering subtypes, we can better treat Alzheimer's disease. So to do that, we are conducting genetic analyses. What you can see here is an example of a genome-wide association study. Boston University is one of the leaders in Alzheimer's disease genetics. We're leaders of the Alzheimer's disease genetic consortium. And we have a cohort of over 70,000 individuals, cases and controls, who have been richly genotyped. We've looked at their genetic architecture and looked at differences. And as part of that research, we've really looked at subtypes, and that's what I'm showing here on the right, that if we can effectively group our folks with Alzheimer's disease into different subtypes, then we might be able to better treat them, and that therapies will be more effective. So with that, I'm going to stop. Thank you so much for listening to my work, and here is some contact information if you have any additional questions.
Thank you, Jesse. That was really brilliant. Always inspiring to see what great work you and your group are doing in both Alzheimer's disease and CTE. We're going to shift now to Ludi Shi, a little bit of a different program. She, again, is Associate Professor of Neurology and the Director of the Deep Brain Stimulation, or DBS program, as we call it here at BU. Ludi?
Great. Thank you, Dave, Dean Antman, and the organizers of this panel. I'm going to talk to you a little bit today about Parkinson's disease in a slightly different flavor, but you'll hear some similar themes to what you have just heard, because we are dealing with neurodegenerative diseases of aging, and we're learning lessons from each other, including folks like Jesse Mez and Michael Osko, as we think about Alzheimer's disease and chronic traumatic encephalopathy. But the twist on this, I'm going to talk to you today about the promise of digital technologies to aid in the diagnosis and management of diseases like Parkinson's disease. So what is Parkinson's disease and Parkinsonism, and why are they important? Well, I think that many of you may know that one of the most public faces of Parkinson's disease has been Michael J. Fox, among many others. Here's a picture I just absolutely adore. He's here with his co-star from that very famous Back to the Future movie. And you can just see the concern on his face as he sees Michael now entering his 4th decade of living with Parkinson's disease. He developed it quite young at the age of 29. And like what you heard from Dr. Mez, the number of cases globally is rising. We're currently dealing with 1 million cases of Parkinson's disease in the US, and it's much more globally. And there is evidence to suggest that it is increasing in prevalence as time goes by, in addition to the large increase in our aging population as time goes by. So you know from Parkinson's that a lot of it is centered on motor symptoms such as slowness, rigidity, and tremor, but some cognitive impairment can be an issue as well. And I wanted to highlight that a number of studies over the years have demonstrated that Parkinsonism, the syndrome of the motor symptoms like the slowness, tremor, and rigidity, they're actually not unique to PD itself. In fact, it's been demonstrated that you can actually see these signs in Alzheimer's disease, perhaps in a subset or as the Alzheimer's disease advances. There's an increasing recognition that there's a mixture of brain pathologies that lead to both Parkinsonism and probably dementia. And we now know from traumatic brain injury cases that traumatic brain injury leads to neurodegenerative conditions, including things like Lewy body pathology. And Lewy bodies are a microscopic finding that are characteristic of Parkinson's disease and the related disorders like Lewy body dementia. And now we see them in people who have had chronic traumatic encephalopathy after head injuries. So we're going to learn, I think, as time goes by, that these diseases have a lot to do with each other, and we need to be learning about them as we try to develop therapies for them. Now, I'm going to talk to you about biomarkers. You've heard some examples of them already, but what they essentially are, for those of you who may not know, are any sort of information from an individual that can tell us a little bit about their disease state. So it might be neuroimaging, it might be blood samples, it might be DNA, but I'm also going to tell you that your digital signatures in terms of how you move, or maybe even how you think, are increasingly being looked at as a potential biomarker, a so-called digital biomarker, if you will. Now I'm going to show you an example. The biotech and pharmaceutical companies developing treatments to slow down things like Alzheimer's disease and Parkinson's disease are increasingly looking to technology to try to see if a rich set of data about how a person performs might actually be an accurate measure of how people progress, or hopefully progress more slowly if they're given a therapeutic treatment. And this is an example from a recent clinical trial looking at an antibody against alpha-synuclein, which is a protein that people think is important for the development and the worsening of Parkinson's disease. And the company, in conjunction with other scientists, developed a smartphone app to let participants, individuals with PD, perform a few tasks over time to see how it would compare to the gold standard way that we currently measure Parkinson's, which is actually kind of a complicated clinical scale that trained nurses and physicians do for patients. And it's about 31 tasks that a person has to do, and the score is derived. And the data on the bottom just simply tell us that some of the digital signatures that were captured from just interacting with the phone seem to correlate well with the gold standard way that clinicians actually define how much impairment a person has. For example, with dressing and finger tapping. Finger tapping is basically a proxy for some manual dexterity. Posture and balance are clearly related to each other. And turning around with the smartphone in your pocket seems to correlate very well with an overall degree of measure of how someone performs with respect to their walking. So the reason why is this: in our phones and Apple Watches and in a lot of sensors, we have hardware that lets us determine velocity in rotational directions, but also up, down, and left and right directions. These are called accelerometers and gyroscopes. And they're used in a lot of devices, some of which are research grade, which you're seeing here, but they've been used with very good effect in many natural history and biomarker studies. And they're able to now show us that they can detect signs of neurodegenerative disease very early on. And there are multiple well-regarded publications demonstrating this. But aside from research, I often also think like a clinician, and I think about how can technology actually help us care for our patients better, or more simply, or perhaps more conveniently. As the COVID-19 pandemic has showed us, we need to think of ways to deliver care where it's needed into the homes of people with Parkinson's disease. And this is an example using Apple Watch. Apple Watches are now ubiquitous. They are collecting EKG data and heart rhythm data, but data scientists at Apple have converted the data, raw data coming from those accelerometers within the smartwatch to tell us information about how a person with Parkinson's disease might do over the course of the day. For those of you who are familiar with Parkinson's, you know that quite a bit of medication can be quite helpful for relieving the symptoms of tremor. And they're taken at first maybe just 3 times a day, but as the course of the disease goes on, someone might have to take it several times a day just to keep functioning, just to keep their tremor at bay, to keep their slowness at bay as well. But also, unfortunately, with time, people can develop involuntary movements called dyskinesia. And some of you have even seen this in Michael J. Fox as he sort of wriggles around on video if his medication is really causing this symptom. And so what you see is the Apple Watch generating signals where it's reliably showing the ups and downs that a Parkinson's patient might go through with respect to the dyskinesia they might see, which fade away and then start up again after a person's taken their medication. And likewise, tremor, which may come on as the medication's wearing off, but goes away as the medication starts to kick in. And we are now seeing the power of this data being related to practical clinical use potentially. And so I think that we're getting ready to do a study hopefully later this year in which we may incorporate this in terms of helping manage patients with Parkinson's disease and trying to use information from the person as they're sitting at home, interacting with us in a telemedicine visit, also incorporating data like this from an Apple Watch, such that we might be able to think about titrating their medications, recommending lifestyle changes, and even adjusting their deep brain stimulation from home. And with that, I'll thank you and would like your advice and your questions regarding this kind of work.
Great. Thank you so much, Dr. Shi. It was a really wonderful talk. Our last speaker is Dr. Lan Zhao, who's MD, PhD. She is a professor of neurology and pathology at BU School of Medicine and our vice chair for research. She will be taking us in a different direction, talking about muscle disease. So Dr. Zhao, I'll turn it over to you.
Share screen. Can you see my slides? Okay, great. Okay, thank you, Dave, and thank you, Dean Antman, for organizing this to present our research. So our research focuses on developing drug therapies to reduce inflammation and scar formation in Duchenne muscular dystrophy. Duchenne muscular dystrophy is the most common genetic muscle disease, which affects 1 in 5,000 live male births worldwide. The disease was first described by French neurologist Dr. Duchenne in 1868, so the disease was named after him. The disease is caused by mutation in the dystrophin gene on the X chromosome. The disease causes weakness of limb muscles, breathing muscles, and heart muscles. Duchenne boys usually develop limb weakness, like leg weakness, around age 3 to 5 years. And the weakness worsens over time, so the Duchenne boys will lose ability to walk and become wheelchair bound by age 12 to 20 years. And they usually die by age 20 to 30 years from breathing muscle weakness and heart muscle weakness. So the disease is fatal. I'm a muscle doctor. I'm dedicated to the research to find the therapies for treating this disease. Here are some pictures of Duchenne patients. You can see that the Duchenne boy, they cannot walk normal. They walk on toes. That's because of the muscle weakness and Achilles tendon contracture. If they are put on the floor, they cannot get up without using their hands to push the floor and pushing their own legs to get up. This is because the weakness of the proximal leg muscles and the pelvic girdle muscles. They often show big calves, although the calf muscles are very weak. And the bigger calf is not due to increased number of muscle cells. This is because of increased the scar tissue within the muscle. And this is the muscle biopsies from patients with Duchenne muscular dystrophy or Duchenne muscular dystrophy mouse model shows prominent muscle inflammation. See, and the left panel is the H&E stain of the muscle section without muscular dystrophy. The right panel shows the H&E stain with muscular dystrophy. You can see lots of small blue cells, and these cells are inflammatory cells. the inflammatory cells, they can cause damage to the muscle. They also induce the collagen-producing cells to produce excessive collagens, leading to scar formation, because collagens are a major component of the scar tissue. And the collagen stain shows that in the normal muscle, there is a limited amount of collagens and separating the individual muscle cells, providing the structural support to the muscle. But in patients with Duchenne muscular dystrophy, the collagen deposition is dramatically increased. It's like lots of scar tissues in the muscle. And these scar tissues can impair the muscle function. Muscle cannot contract or stretch well with this amount of scar in the muscle. So people did study showing that the degree of scar formation, scar tissue in the muscle, directly correlates with the weakness of the muscle. Currently, there is no cure for Duchenne muscular dystrophy, but the therapy development is very active, mainly in the 3 directions: to develop gene therapy, cell therapy, or drug therapy. Since the disease is genetic, so people try to develop a gene therapy, put the normal dystrophin gene back in. However, this gene is huge. This is the largest gene in human, which is beyond the carrying capacity of viral vector. People usually use the viral vector to carry the gene into the cell, but the gene is too big. And people develop a microdystrophin gene to delete some parts within the gene, but the microdystrophin is not fully functioning. Then people try to develop other advanced technologies, for example, exon skipping technique to skip the area with mutation. And so far, there have been 3 exon skipping therapies approved by FDA, which can skip exon 51 or exon 53. But this technique, this therapy, can only help 21% of the patients with Duchenne muscular dystrophy. Scientists are actively developing genome editing therapy to correct the mutation within the gene. 60 to 65% of the patients with Duchenne muscular dystrophy are caused by mutations. It's a large deletion of the gene, not like a single nucleotide mutation. So these advanced technologies do not help these 60 to 65% of the Duchenne patients. And we do need better therapy to help majority of the patients. The major hurdle of the cell therapy is the cells, the stem cells, they can get into the muscles, but they cannot survive very well because the muscle tissue is not normal. It has lots of inflammatory cells and extensive scar formation. The local muscle environment is very poor in muscular dystrophy. So our research is try to develop drug therapy to reduce muscle inflammation, reduce scar formation, to improve the local tissue environment so that we can improve the muscle function, prolong the survival of Duchenne patients, and also to facilitate the gene therapy, cell therapy, so that the gene and the cells can get in, stay, and function. We have made significant contributions to this research field. First, we improved the mouse model for Duchenne muscular dystrophy. To test the therapy, we need to do preclinical trials. People usually use a disease model for that, and we improve the mouse model. In this mouse model, we also characterized the progression of inflammation and scar formation so that we can target the timing, to test the therapies. We've also did proof of concept study demonstrating that reducing inflammation and scar formation represents a useful therapy for Duchenne muscular dystrophy. Currently, we're actively exploring the individual cells within the muscle, which are involved in the inflammation and the scar formation, and try to develop a more targeted therapy, safe and effective to reduce muscle inflammation and scar formation. By doing so, We hope to prolong the survival of Duchenne patients and improve efficacy of gene and cell therapies. Thank you.
Great, thanks so much, Lon. We really appreciate it. That was a wonderful talk and great to hear. I encourage you to continue to put your questions into the Q&A. We have several questions. I think we have one last slide to show, Nick, if I'm not mistaken, if you can put up our departmental slide. Love to show this slide. A, because nobody's wearing a mask. This was pre-masks. B, because it really shows the great diversity of our department. We have over 60% women on our faculty, which is pretty much unheard of in neurology. We are also very committed to diversity from a racial standpoint and really from every standpoint. The diversity is what makes us truly great for everything that we do. Very proud of the department that we've become. Everybody's smiling, not just for this picture, but we smile a lot all the time because we really love our jobs. We love working at Boston University. We love taking care of the patient population that we do. And we feel very privileged to engage in the research that we have going on right now. So I hope that this has thrilled you as much as it has me. I'm going to get to the questions. Maybe Nick, you can take down that slide and we'll have all of the speakers, please show yourselves and I will direct questions to you. First of all, to Dr. Alasko. Someone wrote, when do you think diagnosing CTE by MRI or other parameters will be available to the public? Great question.
It is a great question. It's one we get frequently. I used to try to put a number on it and I learned my lesson pretty quickly on doing that. But I will say, so it's hard to say when exactly, but I will say the next year or so, I think there's really exciting developments that's happening on a lot of different fronts. So one, actually Dr. Mez and myself have, we think, a really important paper that's being reviewed currently. And I think it'll really move the field forward in terms of MRI patterns in CTE. There's also, new criteria that are currently, soon to come out. Hopefully, these are really research criteria, but again, they're meant to inform in the future clinical criteria as well. As I was talking about with the PET imaging, we're learning more and more about the tracers and how they behave in CTE, what works, what doesn't work. And we have funds to carry us through for the next two or so years, and hopefully we can get more funds to really accelerate that development. So I'm really hopeful, though, I said in the next couple of years or so, we'll have a really good idea and made a lot of progress on that front.
Great, thank you so much. I'm going to ask the next question to Dr. Mez. Are the interviews with family members intended to identify patterns in behavior?
So yes, absolutely. So the interviews are very much like what we would do in a traditional memory clinic. And what we're really trying to do is identify the mood, behavior, and cognitive symptoms that occur in folks with underlying CTE pathology. So the things we look most carefully for are symptoms of depression, symptoms of disinhibition, symptoms of having a short fuse, behavioral dysregulation. And we think that those may be related to CTE neuropathology.
Great, thank you so much. I'll direct the next question to Dr. Shi. We have a patient who was diagnosed with MSA. Can you tell us a little bit about what MSA is and any trials that are going on in that?
Yeah, that's a great question. So MSA stands for multiple system atrophy. It's what sometimes is referred to as one of the atypical Parkinsonian syndromes. In fact, it does have something in common with Parkinson's disease aside from just the symptoms. There's thought to be, again, that alpha-synuclein I mentioned is a protein that seems to be also showing signs that it's a toxic player in multiple system atrophy. It's not altogether clear. It does have a slightly different sort of pathology, but it is a more progressive condition in a lot of cases. And it's gathered a lot of interest, and there are a lot of clinical trials going on as people realize that this is a real devastating disease that we need treatments for. Luckily, there are also treatments being developed to treat a lot of the symptoms of multiple system atrophy. So we're seeing some signs of real progress there.
Great, thank you. A question back to Dr. Alasko that I think is on the tip of every parent's mind. What percentage of high school and college football players, and I would throw into that soccer players, lacrosse players, probably you're only safe if you're playing badminton or bowling these days. What percentage are likely to develop CTE, and how readily can one suspect clinically silent early CTE?
This is actually a great question for Dr. Mez, but I will take a stab at it too. So I think this kind of gets at more prevalence of disease, something we don't know a lot about CTE. We think it's common, but we don't have great data to provide direct numbers on prevalence. Dr. Mez has published a paper that talked about, in our brain bank at least, that the proportion of people in our brain bank who played high school and college and who had CTE, and the college football players is around 85 to 86% or so. And I think in the high school players, it was around 40 something percent, estimating. So we do think it's common, and we really need some of these more epidemiological studies to identify the true prevalence, and hopefully the Boston University football study that Dr. Mez is referring to will help us get us there.
Great, thank you. Last question, I'll send it back to Dr. Mez unless you want to punt it over to Dr. Alasko. Abbott just announced preliminary approval of a blood test for mild TBI. Can you please comment on that?
Yeah, I'm probably better to answer that than Dr. Alasko. Right, so that is a test for acute concussion or acute TBI. And what that really tells us is it's a nice way of ruling out changes on a CAT scan. If somebody has a potential head injury, or they're not sure if there was a head injury and they come to the emergency room, if this test is negative, it means it's unlikely that they'll have changes on their CAT scan. That is very different than what we talk about when we're thinking about chronic traumatic encephalopathy, where we think it's hundreds, thousands of hits over somebody's lifetime while they're playing football or hockey, when they might not have any changes on their CAT scan, but it's this accumulated burden of hits over years that we think leads to this disease.
Thank you. We'll do one last question, then I'll wrap it up so we can end on time. Is there a gold standard to diagnose dementia? Does lumbar puncture help? Anyone want to take that?
Mike, do you want to do this one, or should I?
Dementia, we can split it. So dementia is not a disease. It's actually a clinical syndrome. So it's kind of like a fever. It tells you something's going on. So dementia is defined by problems with memory that interfere with your ability to function. And so there are clinical criteria, established criteria to diagnose dementia. What we use is things like lumbar punctures or imaging to help us figure out the etiology. So is the dementia from Alzheimer's disease? Is it from Parkinson's disease or CTE? And it's important to also know the etiology because it has important treatment and diagnostic implications.
Thank you very much for that eloquent answer. I want to thank everybody for joining us today. This panel has been recorded and will be made available online. And please do feel free to reach out directly to any of the investigators you heard today. This panel allows us to share the department some of the research. There's more that's being done that's very exciting as well that we're happy to share with you. It also serves as an opportunity for us to interact with the broader BU School of Medicine and BU Medical Center community of alumni, the faculty and staff, the students, parents, friends. There are a lot of you out there, which we really appreciate the ability to connect with you today. Our audience today is joining us from all over the country and perhaps all over the world. We really appreciate you taking the time with us. While this format has allowed us to address your questions, we really encourage you to send any further questions or interest in our research. Either reach out to the panelists or to any of our staff in development. You can email BUSMDEV, that's for development, at bu.edu. That's BUSMDEV at bu.edu. It sounds like I'm talking in letters now. And we'd be happy to connect you. You can also reach out to me directly. I'm happy to direct your questions to another topic that maybe wasn't covered today as well. So with that, I will wrap it up and thank everybody. Thank you to all the panelists and to Dean Antman and the Development Office for putting this together today. And thanks to all of you for joining us. Really a wonderful seminar. Thanks so much.